Stone, who's director of a food acceptance group, plus a few other titles, I believe, at Stanford Research Institute, and his colleague, Dr. Joel Sidel, who will tell us about descriptive sensory analysis that they have developed at Stanford Research Institute. And I gave you some descriptive material prior to this lecture, which I hope you've had an opportunity to read. Dr. Stone?
Thank you. Now that we've gotten all the descriptions over with, what I want to do is, or what we plan to do, the two of us, is talk to you about descriptive analysis as a sensory procedure. And in talking about it, we're going to, if you wish, take you through some rather simplistic things in the beginning and then sort of build up to more complex aspects of sensory evaluation. And I will talk to you for most of the lecture, and then my associate Joel will get up and finish the rest of the lecture. And let me begin first by showing the first slide, which somehow will now appear magically. What the first slide shows, is a very simple statement which goes as follows: that sensory perception is an integrative function of the brain. We use our senses, vision, audition, taste, smell, touch, pain, temperature, to evaluate the attributes of a product. Product can be a material, it can be a beverage, it can be wine, it can be toothpaste, it can be the hardness of the chairs, the softness of the pen, the colors, etc. What we're talking about is an integrative function of the brain. And to further expand upon that, this is just a simplified picture of what it is. We're dealing, as I said, with many different senses. Now, in terms of sensory evaluation itself, we're using sensory tests as a model to predict consumer behavior, or at least to have a better understanding of the consumer, or the consumer's behavior relating to a particular product or group of products. We have panelists who hopefully reflect the target population. That is, the people who will purchase and use the particular product. We have the test products, then we have production of the product, then we have, in the case of the panelists and the test product, we have a response measurement. And of course, ultimately, in the consumer end, we have the acceptance of the product. So what we're doing basically is using the sensory test as a model, using panelists evaluating a test product to obtain measurements about that product or group of products. Yeah, that's the wrong way. Well, here we go. One more. That's in case you missed the first 4 slides. Is that clear? Focused? Now, what I want to do, as I said, is we started very simply, and now we're going very fast. We're giving you the whole course in 1 hour and a half or something like that. You'll be all finished. When we talk about a model for predicting consumer behavior, we have to go from that model, let us say, down to how to do the actual testing. For example, there are a limited number of testing techniques that we can use to improve product successes or success. We can do an acceptance preference test. Does the consumer like our product? Will he or she buy it? We can do quality judgments. What product defects are present and to what degree? Or we can do a discriminative test. Are selected products detectably different from each other or from some standard? And finally, we have descriptive tests. And these are designed to respond to such questions as, what are the perceptible sensory characteristics of a product and their intensities? Now, in terms of these kinds of tests, I think you're probably all aware that it is relatively straightforward, for the sake of this discussion, let's agree to that one can do acceptance preference tests or make quality judgments or obtain discriminative judgments in a fairly straightforward manner. I'm sure that in reading the text for this course or some of the ASTM publications or related documents, you're well aware of some of the fairly standardized procedures that one can use. And the area where you find the greatest fuzziness, if you want to call it that, or the greatest amount of controversy of a more basic nature relates specifically to descriptive analysis, or describing the perceptible sensory attributes of a product. Now, in order to, let's say, get involved, let's say, in the testing itself, whether it is in descriptive testing or in any other type of testing, but especially in the descriptive testing, you have to concern yourself with a number of problems. For example, do you have a definition of the overall product objectives? What kind of a product are you attempting to develop? What sort of attributes do you want? And related questions. The second one has to do with an accurate definition of the specific problem under consideration. Can you see, everybody see that?
Make it easier for them to take notes.
You can see the slide. Can you see the slide?
And finally, there are questions of the knowledge of consumer test methods. Now, you may assume, or you may quickly jump to the conclusion that these are all very well understood factors related to testing situations. But I think it's very important, we want to stress these because time and time again when we get involved in working with a company on a problem related to product evaluation, typically the first questions you ask are some of these, and typically the answers to all of them is a blank stare. I think it's very important to understand that doing a test for the sake of doing a test doesn't necessarily accomplish anything. And it's very important, regardless of the type of testing you're doing, that you have a pretty good understanding, or at least you've asked the questions and obtained a satisfactory answer to these kinds of questions, because they're very important. Now, they're especially important when it comes to such areas as descriptive evaluation or descriptive testing of a product. Because here you're dealing in an area which is not simply a yes-no type of answer. You're dealing with a more complex problem, which is attempting to define the attributes of a product, the perceptible sensory attributes of that product. And as we say here, the objectives of descriptive evaluation are the first, to identify product characteristics; 2, indicate order of perception; 3, determine subjective intensity for individual product characteristics; and 4, provide a measure of overall impression. So that in descriptive testing of a product, we're very anxious to be able to provide responses or to meet the objectives that we have listed here. Now, I assume that all of you have some familiarity with such methodologies as the flavor profile method. Now, another point I should mention is that if any of you have any questions, don't hesitate to interrupt. Rather than save them up for the end, you might forget them. If you've got a hot question or a hot idea, just sing out. Well, okay, now, the principal applications. So far, all we've said to you is that there are different kinds of testing methods that you can use, that some of these methods are pretty well defined, difference tests, etc., quality tests, quality judgments. And we do those tests with specific objectives in mind. At the same time, we also have specific objectives with testing such as descriptive analysis. Now, what are the principal applications of descriptive testing? Well, first, you have such things as new product development. You're developing a new product from an idea, from a concept. You want to correlate this with instrumental measures or consumer acceptance or both. B, you want to match this product with some competitive product that is now in the marketplace. Or two, you have some quality control requirements. For example, the storage stability of your product. How long a time period can you store this product? What is its shelf life? B, you may want it as a permanent record, a sort of fingerprint, if you want to call it that, or a profile for want of a different term or another term. You need a permanent record of the attributes of that product. For those of you that have worked in an industrial environment, you know there's a sort of practice that goes on. You develop a new product, and the product goes out into the marketplace, and it's successful, and the consumer likes it, and sales are doing very well. And as soon as that happens, purchasing and production and marketing come in and say to R&D, now let's make it cheaper. And so then begins the process of changing the product, but not changing it. The fragrance is too expensive. Let's get a cheaper fragrance. The protein is too expensive. Let's get a less costly one. And so the game goes, and so the product always changes. And if you don't have a permanent record of what you started from, it's possible to end up where you don't want to be, which is a product whose sales promptly drop off, and then you're trying to recall from the formulations as to what it was before. So you need a permanent record. And finally, for troubleshooting, if you have a problem, we recently did a study with a company on these products called Hydrox, the cookies with the filling. Oreos, okay. And it has a cream filling, and this client was very interested in matching the competition. And they prepared a series of products to supposedly match the competition, and we did the evaluations for them and told them where to make the changes. And then they proceeded to make the changes, and before we let them go out into the marketplace, we made sure that they came back to us and to verify with the panel that in fact what we said they should do and what they said they had done in fact matched with what we wanted them to do. And we found that there was something wrong with some of the data. There were some inconsistencies in the filling part of the product. And we went back to them and said there's something wrong with our data, show some changes in this product, especially in the filling. And it wasn't with all the samples. And then we found out that in the process of making the product, they made the filling in a batch, mixed it up and filled half the product, and somebody took off for lunch and waited an extra 2 hours for a long lunch, then came back and started filling the rest of the cookies. And that difference allowed a settling of some of the ingredients in the filling which showed up. So they had a problem of making, somebody forgot something in the process, and therefore it showed up, and this is why you have to match and have a permanent record so you know that if a product changes, you'll know in what way it has changed. Okay, now I want to talk briefly about some of the differences related to profiling and descriptive analysis methods in general. And I want to do this because we're soon, very quickly in this lecture, going into data handling and data processing and scaling, and we're going to talk about scaling methodologies. And I don't believe it's humanly possible to talk about research in psychophysics of scaling. And I think that for purposes of this discussion, I want you to believe me when I tell you certain things. I know it's hard to believe people, but you're going to have to. And for your homework, if you want, you can read such things as Stevens' chapter on mathematics and measurement in Handbook of Experimental Psychology. And there are a related number of papers on scaling. There's a review by Ekman and Schoberg in the Annual Review of Psychology, 1965 or 67, something of that order. And there are a number of books and references that I'm sure Rosemary and Maynard Amerine can provide for you on scaling. But what I want to talk about is that historically, there's always been a lot of difficulty in describing product attributes. And when the A.D. Little Company, which is a competitor of ours, so we're not going to be that nice about them, developed the flavor profile method, there was a tremendous amount of interest in the methodology because it promised a great deal, and still does promise a great deal, about defining the attributes, the perceptible sensory attributes of a product. And many companies proceeded to become involved in this method and the methodology, and there were people traveling around the world teaching everybody the methodology of flavor profiling. And flavor profiling is basically a method in which people sit around. I don't mean that facetiously, they do sit around the table, and and they talk about a product. And there is a person who is a group leader, and they together talk about the product. This is a sort of what you might call an introspective analysis. An introspection is a form of psychological measurement that was popular in the teens and 20s and eventually died a happy death because for a variety of reasons. And what it was is that you merely sat, and if you want to call it rapped about whatever turns you on. In this case, it would be a particular product, be it a cigar, be it a wine, be it anything at all that you wanted to talk about that had perceptible sensory attributes. And you sat around, and out of this you evolved a language, a series of words which described the product, the sequence in which these perceptible attributes appeared in your mind. And the group leader would then bring in various samples of things and spike samples, etc., to familiarize you with a musty smell and with a wet dog, etc., etc., etc. And the method then proceeded on the following basis, that you sat around this room, around this table, and you tasted the product, and then you filled out a kind of a card in which you listed the various attributes, the order in which they appeared, and you used what we call a category scale. Are all of you familiar with what a category scale is? There are a series of categories, and you must assign your judgment to one of these particular categories, not in between. You must categorize your judgment. And the situation was one in which the group leader worked with the people, and they all made a judgment. And then out of this, you could generate what they, I don't care, call it a sunburst of some, which would give you this wonderful sort of like the sun rising in the east or the west or whatever, and these were the various attributes and their relation one to the other on a sort of unidimensional presentation. Now, the method was designed in such a way that you could not subject the information to statistical treatment. Now, it is claimed that you can convert analog digital, you can convert the words, and there is a sort of data processing method that they say that you can use, but they will not claim very much for it because the way the method evolves, introspection does not permit analysis, statistical analysis of one's feelings. Okay. Now, the flavor profile method is still used, and many of you will go out into industry, whether it's the food or some other industry, and you will find that your company, in fact, has been using flavor profiling. And it's sort of a byword in the business. We are the underdogs, and they are the leaders. They've been at this for 25 years. And there's a great deal of controversy surrounding flavor profiling. And the controversy that surrounds it is associated with such factors as mathematical treatment of the data, the manner in which the information is generated from the individuals, the group judgment, etc. And there's been a lot of controversy, I keep telling you that, and there is, but there is controversy because of these problems. Now, the result has been that people have suggested developing alternative methods to flavor profiling. For example, there is a technique of dilution method, whereby one merely utilizes basically the same kind of a procedure, and as you taste the product, for example, then you begin to make dilutions of it till eventually certain sensory attributes are extinguished, i.e. you can no longer perceive them, they're below your threshold, and so other things stand out more readily. And there have been variations on this method of profiling. Now, what we have done is made an assumption, or I should say I started some time ago when I was involved in finishing up my dissertation up here at Davis, and Rosemary and I would often get into long arguments, with the two of us trying to argue with people from Arthur D. Little relating to flavor profiling, and we never could resolve them to anyone's satisfaction. And the university here has done a lot of work on flavor profiling, and I guess you've sort of given up on it? Or still? Now, I took the approach of descriptive evaluation of flavor profiling. I took the approach that there must be a better way of dealing with the problem. And so I started to look into the problem. The biggest argument you get into is that all the people have to agree together. You get a group judgment. And I felt that there had to be an alternative method. Why not take people and go into booths and evaluate the product on this basis? And rather than use a category scale, use some other type of scale. For example, a scale on the right here, which is called, just for want of a better term, is another type of scale, is semi-structured in the sense that you've got some lines there which, with some terms, slight, moderate, strong. Now, we began to work with this particular scaling technique because there was a considerable body of literature in the psychology field, in psychophysics, relating to scaling. And I'm sure you're all familiar with magnitude estimation and the work of Stevens and his group at Harvard over a 35 or so, 38-year span, in this whole question of measurement, of judgment, and the measurement of responses by people. And basically what he found, and what others have since confirmed, and what they have all found, is that you can obtain from an individual consistent, reliable judgments about the perceived intensities of a particular attribute by asking the right kind of questions. And he found that as you went up in level of sophistication from simple yes-no answers to category scales to such things as ratio scales, that you could get more accurate, more consistent judgments from individuals. And also, because people use numbers differently, that if you did not put numbers down and ask people to put a mark off a particular number, but you let them generate their own number, that this was a fine difference. It was a small difference, but a very important difference. Because when you evaluate information collected, for example, via a category scale versus, for example, a ratio scale, you see much better information coming from the ratio scale because a person generally is internally consistent in his judgments. And I don't know, in the laboratory, have you done a magnitude estimation experiments? They have.
Yes,
Okay. See, it doesn't make any difference what numbers you use, as long as you are internally consistent. By internal consistency, I mean, for example, I'm a great lover of 10, 20, 30, 40. So when I'm in a psychophysical experiment measuring relative intensities, I go 10 or 100. If it's 10 times as great, and if it's a tenth as great as 10, I give it a 1. Well, some people go 1, 2, 3, 4, 5, 6. They use those numbers. And other people use other kinds of numbers. But generally, generally you find that a person is in what we mean by internal consistency is once you stick with a set of numbers, you tend to follow those numbers and use them in judging intensities of items. Everybody follow what I'm saying? I hope. Okay. And so we concluded from doing a lot of experiments, to make a long story short, that if we utilize this kind of a scale, a category scale, while we could make discriminative judgments, we needed much greater differences between products to make judgments, to make statistically significant judgments. And also that the variance associated with this type of a scale was much greater. And also there was another aspect of the category scale versus a ratio scale, and that is that in a category scale, the assumption is that you have equal intervals. And we know from a lot of experimentation that in fact that is not necessarily the case. That the difference between slight and moderate may not be the same as the difference between moderate and strong. And that when you use the scale, for example, such as a ratio scale, type of scale, and there's another example, I think, on the next slide, which is a simplified and the academic, precise version. This is a ratio scale. That when you used, for example, a ratio scale, you found that people could discriminate more reliably. Variances were much less. Now, you might assume with a category scale, for example, where you've got a series of categories, you would have much less variance, and that a person on a ratio scale couldn't possibly remember. The way you use this is you merely put a line, in this case a horizontal line across the vertical line at that point which best reflects your judgment of the relative intensity of that attribute. People wouldn't remember, or rather would be all over the map in terms of using this scale. But in fact, just the reverse is true. That when people use this type of a scale, they tend to be very good in terms of the amount of variance associated with a judgment is relatively small compared with the category scale where you've got to put your judgment in a particular category. And if you think about it, people, number one, do not like to be categorized and do not like to be buttonholed, do not like to be pigeonholed. And they don't like to be forced into slight versus strong or moderate. As you can recall, if I get back to the last scale. Yeah. Maybe you want to be in between, and maybe the words slight and moderate mean different things to you than somebody else. And we know that words have different meanings to people, and so therefore we found through a lot of trial and error and a lot of studies on many different products that by using this type of a scale we get much more reliable, accurate judgments from individuals, and that it facilitates obtaining information from people about the attributes of a product. Okay? Now, you may recall, you can't read it, forget it. I'll tell you where you can read it from someplace else. It comes out of, I think, Rosemary, out of your book. It has to do with a group judgment. Now, you recall that I mentioned to you that there were various types of descriptive methods, and there was a lot of controversy associated with flavor profiling. And is that better? No. Oops. It's got a fine touch. How's that? Is that better? Now, one of the most important aspects of profiling, and the other one is the scaling method, has to do with group judgments and a group of judgments. In flavor profiling, everybody sits around, and with the group leader, they decide upon the attributes of the product collectively. And then when they evaluate the product, they evaluate it collectively to obtain the single judgment. And it's interesting that when Arthur D. Little was promoting the profile method, they had a conference on this whole profile method, and there were two papers in that book which, if they had any sense, they would have never put them in. One is the article by Noel Jones, Professor Jones of UCLA, talking about psychological measurement. And the other one is an article by Richard Hall, who's now a vice president with McCormick. And the article by Jones discusses psychological measurement, and he came out with this statement in that book, in his chapter, and it goes, it says, a group judgment is by no means the same as a group of judgments. At best, a group judgment may be expected to reflect unconscious signaling, and at worst, as when discussion is allowed, will result in the domination of the results by the loudest or most assured judge. During the hypothesis-finding phases of investigation, this stricture need not hold. But if one wants valid judgments of, say, differences, he will be well advised to group his judges only statistically, not in reality. And the whole chapter is a very interesting chapter, and yet it sits there in that book, which I'm sure you're familiar with, and it's in your reference library. The other article that I referred to is one by Richard Hall and discusses scaling methods. And in there is a discussion about some scaling methods that he was using, including the semi-structured ratio type scaling, where he was getting very interesting, very accurate, rather reliable judgments about product attributes. But he didn't know how to deal with it, and he wasn't familiar with the scaling work being done at Harvard. And so the result is that it just sits there, and it's a beautiful article, and it's one of the basic articles that we used as developing the descriptive analysis method that we've developed. And there it sits in that book, and it's an interesting book to read now, knowing what we know. It's sort of comical if you think about it. It also points out the rather, being very impolite, very shallow, not very far-reaching thinking of the people who use the flavor, or develop the flavor profile method. Because in there they raise some very serious questions about scaling and about the group judgment. And those are two very important critical issues. Okay, now, wait a minute. Let me go on to the next thing. Okay, now, so what we have done then is we developed a method which goes as follows. We have a product whose attributes we want to define. And this product can be and has been beer, wine, cigars, toothpaste, floor polish, furniture wax. It makes no difference. We've worked with all of these products, and it's not the product that causes the big problems. Well, it does to some extent. But what we have done is we take a product, and we get a panel of people together, and we train them. We subject them to some sort of screening. The screening can be a routine procedure like doing triangle difference tests to determine what we're trying to do in this procedure is to select out people who cannot discriminate differences. And the ones that are left who can discriminate then represent the core group of our panel. Then the next stage that we go with, and typically we start with twice as many people. For example, doing descriptive analysis, we might use a panel of 10. We'll start with 20. And we've got 20 people who are all capable of doing psychophysical testing. They understand how to do a difference test. They know how to fill out a scorecard. They know how to taste the product. They know how to smell the product. They know how to feel it, or whatever else you want to do with it. We then bring them together in a room with a round or square or whatever table that they can all fit around. And if we have more than 10 at a session, we often run separate sessions. You get 20 people in a room, and the social interaction far exceeds anything else. And what we then do is have a panel leader, if you want to call it that, work with them by explaining to them the product and telling them what we're trying to do. That is, we want to define the attributes of this product. How would you explain what you feel, or what you smell, what you taste when you feel, touch, or taste, or whatever this product? And what happens is we get a group discussion going. And out of this group discussion comes a language. This is not very much different from flavor profiling, except for one thing. In our operation, in this type of descriptive analysis, the panel leader does not contribute data. The panel leader's function is solely to help people verbalize the product. If they have trouble understanding what is bitterness, then it is his or her function to go obtain some samples of a quinine solution or some related bitter material and say, this is what bitter tastes like. Okay, now, we then proceed to allow these people to evolve a language which, in order of appearance, defines the various perceptible sensory attributes of that product. And this typically might take, anywhere from 3 to 5 or 6 or 7 one-hour sessions, which these people sit around and talk about the product and taste it and evaluate it over a period of time. If you want, you can do 2 sessions a day for many days if you're in a rush, as all industrial problems are crises. But basically the purpose of this is to evolve a language. And then from the language, we then proceed to introduce that language to the ratio scales. In other words, we have a scorecard with some information up here, which is, besides name, date, code, has to do with what they're supposed to do. You taste the product, or you smell the product, and then you fill out the scorecard, and there'll be attribute number 1, and there'll be a scale here, and attribute number 2, etc., down to as many attributes as there may be, as they may decide that that's what they want to evaluate. And their function is only to taste or smell whatever, and then put a line at some point which best reflects their judgment of the intensity of that attribute. And then they go through this. The first couple of times, now all the testing is done in isolated booths. Pardon me for not telling you that, but it's all done in booths. The first 3 or 4 times they do it in the booths, and then they come out and go back into the conference room and discuss their information, and it's put up on the board so everyone can see it. And typically we often tape the sessions for purposes of a record and to keep track of things and to see if there's certain things that have been overlooked in evolving the language. And after a few sessions like that, we then proceed into actual testing. Now, Before you can do the testing, we have to determine if the subjects are adequately trained. And by that we mean, are subjects discriminating differences? And are they discriminating differences reliably? And are they making discriminations between products? So what we have to do is we use, as we state here, we use statistical procedures to determine, number one, subject performance. Because if a subject is not performing, end of game. You must go back and retrain the subject. There's no sense collecting information if a subject cannot discriminate. How can you use the information? Number two, if the subject is performing, you then find out, is the scale performing? I.e., are you making discriminations? Are the subjects making discriminations on that particular scale or that attribute of the product? There may be many attributes. There may be 5 or 6. There may be 15. So you have to first find out, is the subject performing properly? Is the scale performing properly? And if those two are, then and only then can you go back and say, are there differences in the products? Subject performance. As I mentioned very briefly, are individual subjects responding consistently, or is more training required? And this is very, very important. We have observed, and Joel will shortly show you, if I can quickly end. He will show you data from subjects who have been well trained and so-called experts, but when you evaluate their judgments, you find they're not consistently performing. And you always have to ask this question. You always have to be concerned about subject performance, because you cannot talk about a product, you cannot talk about an attribute until you find out if the subjects are discriminating. In this case, we're asking the two questions. Are individual subjects responding consistently, or is more training required? Are individual subjects adequately discriminating differences among products? Two very important questions. Next, the scales. Scale performance. Are individual scales producing consistent results? And we have seen with long, extended training and familiarization and routine testing, and these are the 8 scales that the panel swears are the best scales. And when you go through the data, you find on 2 of those scales the subjects couldn't discriminate anything. So you always have to look at data and look at it very carefully, and you have to follow it in a logical sequence. Are individual scales producing consistent results? And secondly, are individual scales adequately discriminating differences among products? And then finally, product performance. Now we finally get down to what we really wanted to do, which is to find out to what extent the products differ on specific attributes that were judged. Now, I've sort of gone very quickly. For example, I went through scaling. I pointed out a category scale and a ratio scale. Those, by just putting up on the board or showing that slide of the category scale and ratio scale, what I showed you was about 30 years of research progress in psychophysics. Hindsight is always better than foresight. I've got the advantage of all that research, and that alone. Those 2 scales represented 30 years of difference, 30 years advancement in scaling. Now, I've gone rather fast. I'll answer any questions you have, but I think what we might do is now let Joel talk about data. What do the data look like? And maybe you'll get a little better understanding of what we're talking about when we talk about descriptive evaluation. Okay?
I was looking for a pointer because I'm so short I'll never reach it. Well, that'll help a little bit. Okay. Just a couple of things before I go into data. Data tables generally are boring, dull. And if you're going to try and take notes and copy all that data, good luck. You're not going to make it. I just want to get across some principles to you. But before I get into that, there has been Dr. Stone has mentioned, there are many differences in the procedure we use than the procedure that is used by other people, specifically the flavor profile technique. Our procedure is very highly quantitative. Most people, as Dr. Stone mentioned, will go in and just take a look at product differences or mean differences between products on some sort of scaling procedure and say, well, the product is different, yes or no. But we won't allow ourselves that luxury. We go in and we look at individual scales and individual judges. And I'm going to show you how we go about doing that. But critical, of course, to doing that is to have a scaling procedure that allows you to utilize the appropriate kinds of statistics. For example, there has to be a match between the scale you use and the kind of statistics that you're allowed to use to analyze your data. That is why we feel as though it's necessary to utilize the power of parametric statistics, analyses of variance, analyses of covariance, various kinds of techniques. It's critical that we have a method based on scaling techniques that are more than category scales. And more than, I don't want to get into a discussion on that. The Stevens article can straighten that out. But basically, the interval or ratio scales that we kind of advocate, these make some use of the space between numbers. For example, on the scale that Dr. Stone showed you, you could mark any point you wanted. Well, obviously, if you felt as though one product were stronger or twice as strong or half as strong as another product, you could express this via your fractionating of this scale. Well, automatically, that puts you into utilizing scales that are more of the interval ratio type as opposed to the category type. Okay, so now we set the rules straight. I can use the statistics that I'm going to talk to you about because that is very critical to have that proper matchup. This is the kind of data display that we get out from our computer analyses. This represents one attribute, one characteristic. This characteristic is going to be aroma. Now we know that we can have many characteristics, 5, 10, 15, whatever dimensions there are. Each individual characteristic has to be analyzed separately to determine whether the judges are discriminating, to determine whether or not the scale is useful, and then to go in and say, yes, there are differences among the products on that dimension. Now, what we have here, for example, just to describe this, are your subjects 1 through 6, sample A, B, C, and D. We had to take the brand name out. Each individual cell represents replicate judgments. Our technique involves getting replicate judgments. We do not just go in and get one measure from one judge. We'll get replications 10, 15, 16, whatever we feel is necessary in order to generate sufficient data to make reasonable decisions as to whether or not the judge is responding consistently. A good measure of responding consistently is to have the judge measure the same sample on repeated occasions and to take a look at his individual data on each of those occasions. Okay, to take a look at an individual judge, we just take and go one row. Let's take row 1, for example. In this case, what we have is one cell mean. For example, the first one, 12.58, representing the mean of 12 judgments by this panelist. We wouldn't want to present the raw data, the individual data. Wouldn't be able to find a slide big enough to put that up here, or you wouldn't be able to see the print. We run through a one-way analysis of variance on each individual judge. Now, I hope you have had something in statistics. Okay, one-way analysis of variance. Basically, what we're saying is, for that individual judge, is he making significant discriminations? Is he judging the 4 products in question different on this dimension of aroma? Now, we said we went through screening procedures, and we said, fine, if a judge passes various kinds of triangle tests, that we can use him. That's exactly what we stated. That means we can use him to begin training. We have found just invariably, in going in and giving discrimination tests on products of the type we're evaluating. Now, it's different than saying giving a discrimination test on a simple solution of salt, sweet, sour, and bitter. If we take, for example, if we're interested in brownies or cakes or something of this nature, we'll make up samples that are different dimensions and see if judges can go through triangle tests, discrimination tests, and tell the difference between these products. If they do, fine. That still does not mean they're going to function in the descriptive test okay, fine. It doesn't mean they're going to do that. That's why we have to go in and analyze their data as we're working with them to determine if they are in fact trained. Just by passing a discrimination test doesn't mean that they're going to be able to perform on this type of test. It just helps us to reduce the number of judges we're going to have to work with. Since it's a very difficult and time-consuming training procedure, we want to make sure at least that the judges can tell differences between the products in question, or products of this type. The one-way analysis of variance, in this case, as all analyses of variance will give us, we have the mean of all of the samples for that judge, which at this point isn't too meaningful. Standard deviation here, which is really a measure of error, which is error on the judge's part, the judge's variability in his judgments. And we really get to the core of what we're after, and this is the individual F ratio for individual judges. As we go along this row, we see, in fact, some different numbers. And what this really tells us is whether or not the judge is making significant discriminations. All I have to do is go in and tell you that on this bottom line of probability, if a judge has a value greater than 0.50, he's not discriminating. His judgments are pure chance. If he has a value somewhere between 0.25 and 0.50, I'll say he's contributing something to the overall data analyses. But he's not making fine discriminating measurements. If I go in and find a judge at 0.02, 0.00, 0.01, what I'm really saying is that this judge is in fact discriminating differences on those products on that dimension. So this tells me in one quick glance that Judge 4, for example, at 0.93, if I have to do some more training, that's going to be the #1 judge I'm going to work with. This judge is not making discriminations on this particular dimension for these products. That doesn't mean that the judge is going to be thrown out and not used, because we have found, and we'd like to pass this on to you, I'm sure you're aware of it already, that older or flavor profile procedures or older procedures in descriptive analyses made assumptions. These assumptions were that once you screened some judges and trained them, that they were going to be experts on all kinds of products that you were going to use them on. They were it. Made another assumption. Made an assumption that given one product, that on all the dimensions that you're going to make measurements on, by gosh, they were going to be able to do that too, being trained expert judges. Well, we have found that this is not the case. That in fact, a judge may be good on one type of product and may be a bomb on another kind of product. They just may not be able to do this kind of procedure on another type of product. Also, if you have 15 dimensions, chances are you're going to find very few judges that are good on all 15 dimensions. So it's up to you to be able to have some sort of technique to go in and find out which judges are weak on which dimensions. That tells you either to try and train those judges on those dimensions, or it says when it comes to analyzing the data, better be careful on how you use their data, if in fact they can't make meaningful discriminations. In other words, no equal weighting. Each judge on each dimension has to perform.
How are the figures in the last line derived?
Pardon?
How are the figures?
This is simply a one-way analysis of variance with the F ratio, and this is the probability of F being significant. Computed probabilities. Rather than having you run to a table and say, given a certain degrees of freedom, is there significance or not, we save you the trouble. We compute the probabilities right out based on the F. So all this data, that's fine, look at it, but what's really interesting is that final row of probability of the judge making significant discriminations. Okay, what we like to do, what we have to do, is take a look at our judges as a panel. And let's take a look at some data from a whole series of scales, of dimensions, where we just take a look at this last row for each judge. And here what we have is just those probabilities. So let's take a look at Judge 1. What I've done is simply indicated, just by a number count, the number of times, or the number of scales, that the particular judge was at 0.50 or higher, which means that he was not discriminating. Okay? Basically, we can quickly run across and see which judges are having the greatest deal of trouble. We can see that Judge 1 has missed on 6 scales. Judge 5 has missed on 0 scales. So we see that there's a kind of a distribution there. So right away, my faith in the fact that all judges are good in all dimensions is crashed. It's all over with. I know it's not that way. I can also say, well, just totaling up the number may not be fair to the judge because there may be a certain scale that is just nobody's making discriminations on, which may mean that there may not be differences in the product, or it may also mean that that scale is a hard one to work with, that the judges aren't trained well enough on it. Individual data will tell us which of those two alternatives. But we can go over and see, for example, I think we can see, on scale F for the panel as a whole. This column is just for the panel as a whole. The panel as a whole, they are unable to make significant discriminations on scale F. So therefore, if I had a judge that missed scale F, I might not be too concerned with training him on that as an individual judge. The fact is I may have to work with the whole panel, or I may have to consider that scale as not useful. Okay, so this is how we go into looking at individual judgments in terms of discrimination. But that's not enough. Just to say that a panel, in fact a judge, can make discriminations among some products isn't quite enough, because we make certain assumptions in our analyses. And these assumptions are that the judges are going to be, as trained judges, are going to respond in the same way. The kinds of measures we're making are those of identifying characteristics and measuring intensities. We're not asking attitudes, whether like or dislike. Attitudes are a whole different ballgame. We're just asking simple questions of identifying, naming, and getting a measure of intensity. We expect the judges will be a little more consistent here than trying to measure their attitudes of what they like or don't like, what they will buy and won't buy. Going back to the first slide on data, need to focus? Okay, I think we need a little bit of... Is that, well, is that all right? Again, it's really principles I'm trying to get across. If you didn't even look at the slide, if you just concentrate on the kinds of principles I'm trying to get across, I think that would be sufficient. Okay, next thing we have to ask ourselves is the judge evaluating the product in the same way as the rest of the panel? Trained judges should sort of have a good deal of agreement with each other as to which product has the most or the least of a specific attribute. Let's say aroma. Which one has the most and least aroma? Now, in a two-way analysis of variance, which we also do on the data, first we do the one-way on the judges, then we do a two-way analysis of variance of samples by subjects. And whenever you do your two-way analysis of variance, samples by subjects, you also have a term called sample by subject interaction. Now, many people like to ignore that term. Many people, in fact, like to ignore the subject's term and just like to look at the sample's term. And if samples are significant, run off and say, hey, we've got something. Not so. We're interested in looking at that interaction because what does that interaction really tell us? In simplistic terms, what that tells us is if we have a significant interaction, if the panel as a whole shows us a significant interaction, judged by sample, that means simply that the judges do not agree with each other in terms of the samples. Some are rating it high, some are rating it low. What it really means is a deviation from parallel lines. If we were to just take the panel as a whole, if this were an intensity dimension, 0 to 100 or whatever, if we had two samples and the panel as a whole rated sample A down here and sample B there, we drew a line connecting it, that's the panel as a whole. If we took a look at one judge and found that that judge, well, he kind of agreed with sample A with everybody else, but in sample B, no dice. He didn't agree. He deviated from parallel lines. That means he didn't see things in the same way. Now, he may, in fact, not be quite parallel, and that spread is very important to look at, because at least if he's seeing sample B as higher in intensity than sample A, like the rest of the panel, at least his rank ordering of the samples is intact. But of course, the thing that we always worry about is that one. The total disagreement. Now, how do we handle that? There are many ways of handling that. It could be that the judge is confused. It could be that he can't make a measure on that dimension. Or it could be he represents a significant segment of the population that in fact sees things that way. That's important to know. And again, only by taking a look at individual data of the panel, of each panelist, and as the panel as a group, will this be revealed. When you get all these data printouts, there's much time poring over them, but the information that can be gained from it can be very, very useful. In fact, there are certain characteristics, certain dimensions, attributes. You can take something like diacetyl, a term used by the brewing industry. that by looking at individual data may tell us perhaps there are tasters and perhaps there are non-tasters. Perhaps trying to group everybody's data is not too meaningful in a case like that. This type of information, taking out the individual interactions, will tell us that. We have here computed for each individual judge his contribution to the total panel's overall interaction F ratio. The interaction F ratio right here, 1.277. It was not significant at a 0.25 level. But we want to know something about the individual judge. So what if, in a two-way analysis of variance, you get a significant interaction? What about the individual judge? Well, we've taken and divided out each judge's contribution to that figure. And all we do, again, we get very, very quick about it. We calculate the probabilities so that we don't have to go look them up in a table. And we take a look at each individual judge. Now, in this case, the higher the probability, actually the better off we are. We don't want judges who had significant contribution to the overall interaction. We see, in fact, that one judge here, Judge 5, who was a discriminator. But that's not enough that a judge is a discriminator. He also should be in agreement with the rest of the panel. We can tell that. And we see that this judge has not created enough of an influence to have a significant overall interaction, but it tells us we had better keep our eye on him. We had better watch that panelist's data. There are situations, we don't have a slide here, where in fact you'll find that that judge may have, in fact, caused enough of an interaction to result in an overall significant interaction, and that may be the one judge that is disallowing you from using your data because of a significant interaction. When you have a significant interaction, you cannot go in and use the F ratio for sample effect unless you do a little more statistics. Now, basically, how do you develop the F ratio for sample effect? That's the mean square of the error term, usually divided into the mean square of the sample term. Well, if you can't use that, if you can't use the error term because of a significant interaction, that's not my rule, that's the statisticians tell me that's what you can't do. Basically, you go in and test your sample effect over the mean square of interaction. And in fact, if we could take a judge's data out, if we're convinced, in many cases what we'll have is a judge who will be a non-discriminator, like 0.93 there, but somehow or other his judgments will have been different, so different from the rest of the panel that he will have a highly significant interaction. He's not even discriminating, but just by the throw of the dice, the fall of chance, his judgments are sufficiently in different places from the rest of the panel to come up with a significant interaction. If this is the case, we may want to dump him. We may have a legitimate reason to do it. Okay, finally, after we take a look at whether the judge is interacting, whether the panel is interacting, whether the scale is working, or whether the judge can make discriminations, then we get to the whole thing we're interested in. Are those samples different on that dimension? That's really what we're after. But before we allow ourselves the luxury of saying yes, we go through all of that other process. Here is simply what we call, well, let's make it simple. You've used tests after analysis of variance like Duncan? Okay. This is really, for our purposes, our Duncan test. I won't go through the computations for you, but basically what it tells us is if we go to a Duncan table with these degrees of freedom, 264 for means of 4 means, 3 means, 2 means, we'll get the same numbers out that you'll get out. For example, like a 2 mean difference, you need a 2.77 for 264 degrees of freedom. But basically all we have to do is say, hey gosh, look, 6, we can draw a line right around this sample D and say that sample is significantly different from samples B, A, and C, and B, A, and C are not significantly different from one another. That's really